Hello, everyone. This is Dr. Hodonu, and it's nice to be back with you here. And it's unfortunate that we are unable to continue our semester together in person, but we will be able to do a little bit here online. So I'm excited to be back with you. I cannot see you, but I see you in my heart. Okay. Anyways, let's pick up from the scientific method, and the chart that is on the screen right now are the steps of the scientific method, which you guys have seen in lab already. We said a scientific method starts with an observation, which leads to us asking new questions. These new questions can then lead to making an educated guess, or what we call a hypothesis. And the hypothesis will lead to research and experimentation. And the research and experimentation can be analyzed, and the analysis leads us to make conclusions. These conclusions can lead to new questions to be asked, or we can publish and present these findings in a journal or at a conference. And so these are the steps of the scientific method. So a good experiment or a good study also has what we call a control, and I think we've already talked about that in lab. A control are the parts, or control experiments or control variables are the parts of an experiment that doesn't change, the things that are kept constant. So that's the idea of a control variable. And we also have what we call a control group. And a control group is a group that we compare our other groups to. So we have an example here, the hypothesis that says antibiotic B is better than antibiotic A in current use for the treatment of ulcers. So which means we have a group of people that will receive antibiotic B and a group of people that will receive antibiotic A, and we would compare them to each other. But we need another group that we can compare both groups to. That is going to be called our control group. This group receives a placebo. And by definition, a placebo is something or a drug that has no physiological effects, no actual pharmaceutical or physiological effects, but it's just it has psychological effects. And so we're able to compare antibiotic B to antibiotic A to a group that does not receive any antibiotic. Okay, and that's here in this next slide. So controlled laboratory experiments are used to test the effectiveness of a medication in humans. And so here we have a group that gets antibiotic A, a group that gets antibiotic B, and our group that gets the placebo. Okay, and this is what it looks like when you analyze the results. So we can use tables or graphs to analyze results, to compare the groups. So we have a group that received A, a group that received B, and the group that received the placebo, and you're able to compare all three groups using a graph here. And so this is the concept of your x-axis, and this is your y-axis, and the x-axis has the independent variable, and the y-axis has the dependent variable. Okay. All right, I think we already spent so much time talking about this in lab, so we will not go ahead and talk about it anymore. All right. All right. So at the end of a scientific method, you have what we call a conclusion, and we draw conclusions from our analysis and from our experimentation and analysis. And this conclusion can be published in a scientific journal where it is peer reviewed before it's published. It can also be presented at conferences, posters, and presentations. There's also a concept known as a double blind study right here. A double blind study is a study in which both the experimenter and the individuals that are being experimented on are unaware of what groups they belong to. So the double blind study would be a technician does not know what group the patient is in. So it removes even more human bias.